Hola, hola, hola. Está andando. Uy. Sí. Hola, hola. Hola, hola, hola.
Hello, good morning. Can you hear me there? Yeah. For those watching on streaming, please let us a couple of minutes to start, so the people is allowed to come to the hall. Just 2 minutes, please. Good afternoon. This is Enrique Flores introducing our seminar today by Juan Pierella Karlusich. Juan did his PhD in Rosario, Argentina, with Néstor Carrillo, working on photosynthesis related topics. And very briefly, because he's going to introduce himself, then he did a postdoc with Chris Bowler in the École Normale in Paris, working on metadata of the Taras Oceans expedition. That was a great work. And currently is in the MIT doing something more related to structural biology. So, but he will introduce himself. He has just got a Ramon y Cajal grant, and he's interested in our institute to develop his career, and so he's most welcome. Finally, I want to thank Conexión Genoma, a collaborative project in the CSIC genome that has helped us to pay for this seminar, to bring Juan from Boston to here today. So Juan, you are most welcome, and please go ahead with your seminar.
Thank you very much, Enrique. So this is how I organized my talk. I will give you a very brief introduction about my career background. Then I will jump to the title of the talk about photosynthesis and phytoplankton in the global ocean from an ecological and from a functional perspective. Finally, very briefly, my research vision, or a very general overview about that. So, as Enrique mentioned, I did my PhD in Néstor Carrillo's lab in Argentina. Part of my PhD was also carried out in IPK Gatesleben in Germany. But my undergraduate students worked in molecular biology, so that's my background. And after a stay in the Argentinian lab, then I moved to the group of Chris Bowler at École Normale Supérieure, and currently I am part of the group of Sergei Okinokov. We were first settled at Harvard, but we moved recently to MIT. Well, during all these years, I obtained several fellowships to support my salary, and I have 35 peer-reviewed articles, 15 of them as first or co-author or senior author, and I supervise a total of 12 undergraduate and graduate students. So the focus of my thesis was at the end of the photosynthetic electron transport chain. The final acceptor is iron-containing protein called ferredoxin. It is a very susceptible protein to environmental stress. So under such condition, many cyanobacteria and algae are able to express an iron-free protein called flavodoxin. This gene is absent. The flavodoxin gene is absent in the plant genome. So I carried out a study about the evolution of flavodoxin, and I gave support to the idea that the gene was lost during the transition of the green algae lineage from low iron ocean environments to high iron freshwater environments. Because I obtained, I gave support to iron deficiency as the main selective pressure to maintain the gene in nature. And in a parallel project, I carried out the analysis of transgenic plants expressing a cyanobacterial flavodoxin. The protein is still operative in plants, and it confers stress tolerance. So in addition to demonstrating that in crop species like potato, I carried out the description of the main genome-wide responses associated to the expression of the recombinant expression of flavodoxin in plants. Then I jumped to the group of Chris Bowler, where I focus in the mining of the Tara Ocean's circumnavigation datasets. This expedition collecting samples from all the main ocean regions around 10 years ago, and they obtained a high amount, or a gigantic amount of DNA and RNA sequences, as well as millions of confocal microscopy images, as well as physicochemical determinations. At that moment, I carried out studies regarding the global ecology of phytoplankton and nitrogen fixers. I will go deeply about this later. Currently, in the group of Sergei Okinokov at MIT, I am focusing in structural biology. The group of Sergei is one of the pioneering labs that develop these new AI tools that are used to for the prediction of protein 3D structure using the sequence alone as input. And these are very powerful tools. As an example, this is a nice example. The protein at the left has been characterized experimentally its function, whereas the protein at the right is considered a hypothetical protein, and both of them share less than 10% amino acid identity. However, the fold is conserved, and so based on that, we can raise the hypothesis that they share similar functions or the same evolutionary origin. So I am using these tools that are developed by the lab for biological questions, and my main project is about the origin of eukaryotes. Eukaryotes were generated due to the symbiotic association between an alpha proteobacterium and an Asgard archaea. The alpha proteobacterium eventually became the mitochondrium, and the Asgard archaea eventually became the nucleus. So I am using these tools to determine remote eukaryotic homologs in the genomes of the Asgard archaea. We have many new proteins associated to eukaryotic features. But well, as a summary, you can see that I had a transition from the plant size to phytoplankton in the environment to evolutionary studies, from analysis of sequence data alone to analysis of structural biology. So let's jump to the main topic of this talk, and it's about the photosynthesis modulation in the global ocean. So I will talk first about why we care about phytoplankton, and then two different perspectives, from a point of view of the ecological drivers of photosynthesis, and then a point of view of the function of different groups associated to phytoplankton and photosynthesis. So marine phytoplankton is super important because one, half of the global photosynthesis is carried out by them. This implies that they are the base of the food webs. They are very important for fishes stocks. And in addition, they modulate the climate in the long term. This is due to a project called the biological carbon pump, in which, in this project, a fraction of the fixed organic carbon in the surface ends up in the deep ocean, sinks toward the deep ocean, and it remains there for centuries or millennia. Marine phytoplankton covers a wide range of phylogenetically diverse organisms. On the prokaryotic side, the most abundant ones are the Picocyanobacteria Synechococcus, Prochlorococcus, as well as other cyanobacteria involved in nitrogen fixation, such as Trichodesmium. On the eukaryotic side, there are different eukaryotic lineages that acquire the chloroplast in independent endosymbiosis events. This includes diatoms, green algae, dinoflagellates, aptophytes, pelagophytes, cycloflagellates, and many others. And it's important to have an idea about a picture of the ocean, about our current communities or the communities of a few years ago, in order to predict how plankton will respond to climate change. There are many stressors, anthropogenic stressors. One of them is temperature, but also acidification and nutrient contaminations and many others. So we need a baseline in order to see if we will see in the future a community composition shift or a biogeographical range shift, or if there are patterns of acclimation or adaptative evolution. And a nice data set to carry out this analysis and obtaining this picture are those generated by the Tara Ocean expedition. Here we have the map of the expedition that was carried out 10 years ago from the main ocean regions, covering different ocean regions with very different environmental physicochemical contexts. A serial filtration system was used to filter the seawater according to different, for obtaining different samples enriched in different sizes of plankton, and you will see later how important this is. And the generated datasets cover metagenomics, metabarcoding, metatranscriptomics, and high-throughput imaging devices that generated, for example, around 5 million confocal microscopy images and other devices. And there were carried out many environmental, physicochemical environmental determinations. So, as an example of what can be done with this dataset, that most of them are publicly available, I will show you very briefly, or focus in one or a couple of results from these recent papers that is focused in the diatom community in comparison with other phytoplankton groups. So in this project, we ask how important are diatoms when compared with other phytoplankton, and how do their communities vary with the environment from the poles to the equator. For this, we used the approach of 18S ribosomal RNA gene metabarcoding. It consists of the PCR amplification of the region of the 18S gene, and then the sequencing of the amplicons. So the unique sequences are used as proxies, kind of proxies of species, and the read abundances of the unique sequences are used as a kind of proxies of the relative abundance of the corresponding species. And here a summary of the data, in which you can see the biogeography for the main the major eukaryotic phytoplankton groups. Diatoms are widespread, but mainly prevalent at high latitudes, with the color code of the cycles corresponding to the diversity, and the diatoms are especially diverse in the equator, whereas in high latitudes tend to be dominated by a few species that dominate the whole plankton communities. When we compare with other type, the other type of major groups, you can see that the range of the latitudinal range is more constrained, and also in terms of abundance, the other groups are more constrained. This can also be seen here when this is an scheme that corresponds to the read abundance in the different size fractions obtained by the expedition. It's interesting to see that diatoms are overall more abundant than the rest, and the non-diatom phytoplankton groups tend to be detected mainly in the small size fraction, like aptophytes, chlorophytes, and pelagophytes, whereas dinoflagellate, but to more extended to diatoms, they are covering the whole plankton size spectrum. And this is very important because small diatoms tend to be gray-seed, and small plankton in general tend to be gray-seed and recycling through the microbial loop, while larger organisms like larger diatoms tend to sink toward the deep ocean as part of the biological carbon pump. As an example of the type of analysis we can do with this global data set, I carried out the clustering of the different sequences. but they are co-occurrent. So we can define communities of co-occurrent diatom lineages. Some of them, we were able to define 25 of these co-occurrent communities, some of them with higher levels of richness than others, or diversity than others. But it was very clear that these communities vary according to the biogeography, to the ocean. And one important point is you can see in red that the Arctic Ocean stands out as a diatom hotspot with 6 endemic communities. And this is key in the context of the climate change, because the Arctic is the ocean region that with the highest tendency toward higher temperature in comparison to oceans in the equators, as well as in comparison with the Southern Ocean. So you can see this type of approaches as example to do this current picture of the plankton at global level. So and here for these specific slides, I want to highlight that diatoms are confirmed as the major photosynthetic group. We can define 25 distinct communities with clear ocean preferences, and the Arctic as a hotspot of diversity. Oh, sorry. But now I will jump off another work related to methodological improvements, because the previous slides were done with the traditional way of working with genetic surveys. So in the community, we use ribosomal RNAs for analyzing taxonomic composition and communities. This has a lot of advantages. We have very good reference database for examples. But at the same time, we have strong biases for working with this traditional approach. One bias, of course, is that the amplification of the PCR step for amplifying the gene. But other biases are extrinsic problems with the taxonomic marker in itself, because the ribosomal RNA gene can vary between, for example, 1 in green algae to 5,000 copies in dinoflagellates. And in addition, it's very difficult to distinguish when we obtain sequences that are annotated as, for example, unknown dinoflagellates. We have no clue if they are photosynthetic or non-photosynthetic. And it's difficult to cover both prokaryotes and eukaryotes, because we have on one hand the 18S gene and on the other hand the 16S gene. So for this other project, the questions were, can we measure phytoplankton more accurate using DNA from environmental samples? Can we avoid the strong biases of ribosomal RNA gene? And here I came with the idea based on my knowledge of photosynthesis. Why not targeting a specific use photosynthetic functional gene as a taxonomic marker? And after a search, I end up selecting PSBO because, well, PSBO is codified for a subunit of photosystem 2. Is essential for oxygenic photosynthesis. And in addition, it's single copy and nuclear encoded. Targeting PSBO in the metagenome was proposed. I proposed targeting that gene in metagenome as a solution to bypass all the previous problems that I listed. And I validated that gene with the interocean. We have in parallel, as I mentioned, molecular data as well as microscopy data. Here I am showing the correlation for different groups between microscopy in the vertical axis and the 18S gene in the horizontal axis. And below the Spearman row correlations, and here the comparison with PSBO that improves in terms of the correlation coefficient, as well as the relationships are more close to 1:1 when we compare with microscopy. I also created a database of PSBO sequences that is nowadays used by different people of the phytoplankton community. In addition to this methodological improvement, it's interesting to use these quantitative methods for analyzing the ecology of phytoplankton. So for this, I can briefly highlight this pattern that I found interesting. Here I show the cell abundance based on PSBO, the mean across the whole Tara Ocean transect for the different size fractions. And you can see that eukaryotes increases in larger size fractions. And in larger size fraction, we start seeing colonial-forming cyanobacteria as well as cyanobacterial symbionts. So this makes sense. And this is the type of confocal microscopy, but the images that we obtained from the parallel samples in the same samples, but analyzed by imaging. But it was very puzzled when we saw a high abundance of Synechococcus in the larger size fractions that are considered to be free living and one of the smallest cyanobacteria, or one of the smallest photosynthetic organisms. When we check in the images, we start seeing a lot of this imaging in the same samples, in which we obtain a high abundance of Synechococcus PSBO, that are consistent with symbionts of Synechococcus with dinoflagellates, as well as colonies of Synechococcus. This is not the first time this has been shown, but we were able to show that these are more widespread than are very widespread and abundant than previously considered. No, because in general the traditional view is that Synechococcus is a free living organism. So here I am showing the example of the images, the biogeography that we obtain based on that data. But well, the good thing before jumping to the analysis of the images was a good quantification based on PSBO. And for this work, we can, as a main conclusion or main message is that PSBO is a promising new gene marker for molecular-based evaluation of the whole phytoplankton communities. The advantage of being quantitative is also good to start integrating molecular data with other data. And this is a work in which I collaborate with people specialized in satellite data. We use here the satellite data we found, let's say, we were able to integrate the satellite data with the data from PSBO to make estimations of the photosynthetic phytoplankton community in the global ocean. And this is very important in the context of improving biomonitoring tools. Satellite data, of course, is very good because it can expand in space and in time. We think that once this, we think that molecular data is very promising for improving satellite data for biomonitoring. And then in addition to the composition of phytoplankton, I also carried out the analysis more focused in nitrogen fixers. Nitrogen fixers are of course, very important because nitrogen is the main limiting nutrient of phytoplankton growth. And in addition, many phytoplankton species are nitrogen fixers in themselves. Nitrogen being the input of new nitrogen to the ecosystem is carried out by the process called nitrogen fixation. It consists of the conversion of atmospheric nitrogen into bioavailable nitrogen. This is carried out by an enzyme called nitrogenase. And this enzyme is present only in a few lineages, because and this in a few lineages of mainly bacteria and more recently a few eukaryotes that are key to the ecosystem to maintain the ecosystem as natural fertilizers. The study of nitrogen fixation is much more. I mean, the topic of nitrogen fixation is much studied on land due to their economic importance in agriculture, but is much less. We know much less in the ocean, even though the fixation in the ocean is as much as the environment do on land, and very similar to what the human, the industry do for generated chemical fertilizers. So here in this project we aim to tackle the question of who are the main nitrogen fixers in the ocean, which are their different lifestyles, where do they live, and for this, the idea was to integrate both the DNA sequences and the imaging data, because the imaging data was underused and is still underused, but both approaches are complement. With metagenomes, we can tackle species that we don't know how they look, and with imaging data, we can see the lifestyles of the nitrogen fixers. And this is very important for biochemical perspective. It's not the same, the leakage of nitrogen towards the ecosystem. It's not the same if they are free living or they are symbionts. Because what I want to mention before that yes, nitrogen fixers cover a wide range of different lifestyle organisms, including non-photosynthetic and photosynthetic organisms, free living, colonial forming, symbionts, and so on. And the genetic studies are usually carried out targeting the one subunit of the nitrogenase, that is the gene NIFH. So for this analysis, I carried out the mapping of all the DNA sequencing against the NIFH database. And the results were used to improve a machine learning model to use to predict images from nitrogen fixers. I mean, for this machine learning model is the equivalent of doing a blast with sequences. Instead of using sequences as input and output, here I use images as input and output. And the main results from this paper is that we detected nitrogen fixers in undersampled ocean regions, including the Indian Ocean, the Madagascar Channel, the South Atlantic near South America, and part of the South Pacific that are still undersampled. And this is important information for designing new expeditions for targeting these regions in high resolution. And the data from the molecular data and the imaging data were very consistent. And this is the type of images that were obtained through the machine learning model, including the filaments of the cyanobacterium calatrix attached to the diatom ketoceros, the symbiosis between, this is a rare symbiosis between Crocosphaera and the diatom Climacodium. This is let's say the symbiosis between Eucinae and an aptophyte, and this is now considered, due to a recent high-profile paper, this is considered already to be an organelle inside this aptophyte. And then free livings of symbionts that are similar to Calothrix, and symbionts of Richellia inside the diatom Rhizosolenia or inside the diatom Hemiaulus, as well as colonies of Crocosphaera and filaments of Trichodesmium. And we can we analyze the different patterns of environmental distribution and biogeography, but I will not go very deeply, but you can see that there are different clear partition, for example, between those nitrogen fixers that are non-photosynthetic, in which we found them mainly in the Pacific, while others like diatom symbionts in other regions, especially in the diatom symbionts on Trichodesmium in the Madagascar Channel and in the North Pacific, whereas Eucinae or the nitroplast in mainly near South America, as well as one sample in the Mediterranean and near Madagascar and so on. And we postulate in that paper many potential new symbiosis. Of course, we don't have, we only have data for raised hypothesis, but one of the images that we obtained there with high frequency in samples with high abundance of nif-H sequences were this type of images in which there is a chloroplast. This is in red due to the autofluorescence of chlorophyll, and these round granules without chlorophyll. And a German group did another expedition and were able to isolate them and characterize them, and it was a nice example that is a Rhizobia diatom symbiosis. That it was very surprising, and now we are collaborating with them, and we I carried out a more deeply analysis of this symbiosis, and have obtained much more images for them, and they are even more abundant of more, let's say, more studied symbiosis like Rhizosonelia and Richellia. But well, in this part, I want to highlight that the integration of data from molecular and imaging is very powerful, and because they are complement, and we were able to determine new abundant hotspots in the ocean, and also postulate potential new taxa. And well, in all, we found that nitrogen fixers are diverse in covering the whole plankton size spectrum, and globally distributed, but with patchy biogeography. So far, I focus in the community composition, but I want to highlight also the part of the function in the ocean. From the different type of data set we can obtain from the point of view of the function different perspective. On one hand, we have the DNA sequences or metagenomes to see what the metabolic potential, what the plankton can do, and from the RNA sequencing or metatranscriptome, the metabolic activity of what they are doing. So we can infer patterns of, for example, adaptation by analyzing the variation in copy number, of gene copy number, and acclimation by analyzing variation in transcript copy number. And at global level, we can see important abiotic factors that modulate the adaptation, that modulate or influence the phytoplankton. including nutrient deficiency. I mentioned already nitrogen, that is an important nutrient deficiency in the ocean, but iron is, let's say, it can be said that it's the second most important deficiency in the ocean. And then gradients of temperature, and also a depth partition that is associated, for example, to a partition of light intensity and light quality. So one example of for studying phytoplankton from a functional perspective is is iron. So I will start with iron because photosynthesis demands a high amount of iron. Actually, there are estimations that half of the iron of the photosynthetic cell, or half or almost all, can be in the photosynthetic associated to proteins involved in the photosynthetic machine. So here I can show how I come again to the gene of ferredoxin, the last electron acceptor of the photosynthetic chain, and it's an iron sulfur cluster that contains 2 iron atoms, and under certain conditions sorry about that. Let's see. I don't know what happened. Maybe it's the cable connection. Okay. Perfect. Okay, let's continue. So this substitutive, the idea here was to check for patterns of adaptation and acclimation in photosynthesis by studying the substitutive response of the iron-containing ferredoxin by the iron-free flavodoxin. You can see that the data from Tara Ocean is nice because they cover samples with different levels of iron. And here I am showing an overview of the data. In the first row corresponds to DNA from metagenomes, and the second row to metatranscriptomes. In the vertical axis, I am plotting the proportion of flavodoxin in relation to the pool of flavodoxin plus ferredoxin, and in the horizontal axis, the iron. It's clear that in terms of gene copy number, there are communities like in which, like Synechococcus, or green algae, or pelagophytes, or silicoflagellates, in which the flavodoxin gene tend to be present in iron deficiency sites, whereas for other cases, flavodoxin tend to be the flavodoxin gene copies do not tend to vary. But when we see the abundance of the mRNA, there is a clear and general response. This is an average of the whole community. Of course, there can be difference at the level of a species, but in all, when we do this average, in all main phytoplankton groups, we see the substitutive response of flavodoxin in low iron conditions. There is another similar substitutive response in the middle of the photosynthetic chain, which is the replacement of cytochrome CC, which contain 1 iron atom, by the copper containing protein plastocyanin. When here I am showing the same type of analysis. This response is more variable in when we compare the different phytoplankton groups. There are cases like Prochlorococcus, or green algae that tend to rely exclusively in plastocyanin. Other cases like pelagophytes, chrysophytes, or chlorarachnophytes that rely exclusively on cytochrome C6, whereas others like diatoms, Synechococcus, and aptophytes contains both genes and are able to do the response. So this is an example of the study in the environment. These responses with flavodoxin, ferredoxin responsive broadly conserved, and plastocyanin cytochrome responsive variable across groups. But these genes are, let's say, well-studied genes that it's nice to see these patterns in nature, but we know about them. So I want to also show you cases in which for genes that are less known. So I will come back to the first article that I show you. This project was exclusively focused on diatoms, and here are the most abundant transcript families, the top 100 most abundant transcript families in the global ocean for diatoms. And in the first place are light harvesting complexes or chlorophyll AB binding proteins. And this is a family that contains subfamilies that do opposite functions. Some of the families, like family F, are involved in light harvesting, whereas other the families like X are involved in photoprotection. So here I check for patterns that can be related to the different different functions of these subfamilies. For example, the acclimation toward death, or the acclimation, yes, to the conditions in the across death in the seawater column. And it's clear that for light harvesting families are more highly expressed in the what is called the deep chlorophyll maximum, which is in samples that were taken between 50 and 100 meters, in comparison to the samples in the surface that were taken at 5 meters. And then I also check for the comparison between the different size fractions, because according to in the different size fractions we sample cells with different size, and there is for larger cells there is a process that is called intracellular light attenuation. It seems that we are capturing this project with the data, because the larger cells tend to have a higher light harvesting to photoprotection ratio. And yes, we speculate that this is related to the intracellular light attenuation. So these are nice patterns that it can be somehow expected, but it's nice because many of these subfamilies, some of these subfamilies are well studied in the lab, but many others, like subfamily Z, for example, are less studied. So it can give support to the idea that they are, for example, for Z, we can give support that they are involved in photoprotection. Okay, and so this is the conclusions regarding this part. No, the most abundant family among the nuclear encoded RNA is the light harvesting complex with different subfamilies, and we see patterns of according to depth partition and according to the cell size in relation to the functions involving light harvesting or photoprotection. And finally, you can see that among these top 100 most abundant transcript family, there is only one domain of unknown function, DAF 285. Domain of unknown functions are families that contain a protein domain in the databases, but we have no clue about their function. Here we see that this DAF 285 is clearly highly expressed at high latitudes. This pattern of expression is also observed for the other phytoplankton groups. This is a comparison with the opposite pattern for heat-shot protein 90 as a kind of control. And the structural prediction based on using the sequence as input correspond to this shape. These type of shapes are naming solenoids because are repetitive secondary structures. One example of this of solenoid are some kind of antifreeze proteins. So based on the data, we can raise the hypothesis about this, and currently we are working with some collaborations in order to determine experimentally if they can be associated to that function. So the case of DAF 285 is good because I want to highlight that this is just one example of domain of a unknown function. When we see the global genes that are in the global Tara Ocean database for the different phytoplankton group, we see that around 5 or 10% of the sequences correspond to the domain of unknown function, and in addition, around half of the sequences correspond to sequence with no detectable protein domain. And this is what we call functional dark matter. So there is a huge room of for discovery new proteins and functions in this data. One of the I have some successful cases in which I collaborate with experimentalists to determine overlooked functions by analyzing this environmental data and complementing that with experimental support to the RISE hypothesis. So this is to highlight very briefly, or to illustrate very briefly about my research vision, that is that with the ocean, these very global data sets like Tara Ocean, but there are many others, are like a gold mine for determining new functions, and we need to combine both data analysis of this data analysis with experimental validation. I am interested in raising or tackle the question of what are the genetic and ecological drivers of photosynthesis from the pole to the tropics, using these new AI tools to help us to determine the different roles of the genes that are currently classified as functional dark matter. And for that, I think we can use checking the environment for metagenomes, analyze them in silico with these bioinformatical tools, but also these new AI tools, checking in the lab from biogeochemical testing, and using them to then select some genes that can be used to stress responses genes in phytoplankton that can potentially be used for improved crop stress responses. So to do the phenotyping of transgenic plants expressing potential candidates for that. But this is, well, a very general overview. But I want to say that this can have impact in knowing about the adaptation in photosynthesis in the environment, and in parallel maybe the design of stress-tolerant crops. So, well, I want to thank you all of you for the attendance, and the group of Sergey, and especially the program Atrae Genoma that is super important for this transition for my Ramon and Cajal position. Thank you.
Thank you very much, Juan, for this fascinating talk. Now it is open to questions, please.
Thank you. And thank you for this peculiar seminar, quite different of what we usually see here. I have two questions. The first one, you mentioned population segregation in latitude, which is easily explained by temperature and light. My question is, it wasn't clear to me whether there is also a segregation, longitudinal segregation, and if so, what is the cause? And my second question is, when there is no iron, there is a selective pressure for flavidoxin, you said. But then, whenever there is iron available, again, there is a selective pressure for ferrodoxin or iron-containing proteins, but you didn't say why.
Yes, for the flavodoxin-ferrodoxin switch is particular because we consider that ferrodoxin is much more efficient in photosynthesis, and also is in a position of the electron distribution that is key, because if you start, if you have a bottleneck there, you can have reactive oxygen species and so on. So for me, I think that the efficiency is one of the main causes to maintain the ferrodoxin gene, because the gene is also present in all phytoplankton organisms. When you do a survey, there were some claims based on Western blot many years ago that there are some species that do not contain the canonical ferrodoxin, but then when you analyze the genomes, you see them everywhere, independently of the iron in the environment. For the flavodoxin gene, yes, I think that that's the problem, that maybe there is a cutoff between efficiency in the chain and the iron content in the cell, the modulation. Maybe that can explain how it works. For cytochrome C6 and plastocyanin is very different, because copper is everywhere and very bioavailable. There are not so many other copper-containing proteins, so maybe yes, there is a preference for copper in general for that reason. And regarding the patterns of longitudinal pattern, well, in general, we don't see clear patterns longitudinally, because there are more related patterns related with ocean regions that more or less you cannot capture that very well. Then there is here I simplify the view, but then you have the problems of the ocean currents. So sometimes these patterns there are some models that oceanographic models that estimate the connection between the different samples, and sometimes when you take into account that, the correlation improves a lot. Yeah, but in general, yes, for ecologists, they tend to be latitudinal gradients for studying. Yeah.
Any other questions, please?
Just to comment. Yes, ferrodoxin in vitro has shown to be more efficient than flavodoxin. So if a cell can have ferrodoxin. So flavodoxin is a replacement, but ferrodoxin is more efficient. About the plastocyanin and cytochrome C6 couple, you have a measured iron, but maybe you should add copper in this measurement, because the flavodoxin ferrodoxin is regulated by iron, but plastocyanin cytochrome C6 is regulated by copper.
Yeah.
So you have not iron, but you have not copper, you have not plastocyanin at all. So
yeah,
it should be copper added.
That's a good point, Jose. And actually, you remind me that we don't have copper determination, but at least we have some estimation from models that more or less there are some correlation. So I should check for the ratio of copper iron in these correlations to plot to see if there is an improvement with that. Yeah, thank you.
Any other questions, please?
Thank you very much for your seminar, Juan. I have very general question. If I understood correctly, what you propose to do in your future position as a harmonica HAL is to somehow combine your background from your PhD and also the knowledge and experience that you have analyzing some massive amount of data, and you want to propose some hypothesis and validate this hypothesis using other organisms, specifically microalgae and plants. But most of the microorganisms that you have shown are not easy to work with, not genetically. It's very difficult to modify genetically and so on. And so my question is, you have some species in mind of microalgae or plants or to validate the hypothesis?
Yeah, that's a good point. I think that the easiest way for me that I plan to do is to focus in model organisms like Phaeodactylum for diatoms. I think diatoms are the good. I like diatoms because they are, when you study in the ocean, they are everywhere, and so you can have them in a different extreme environment, and then we can focus in a model organism. For now, yes, Phaeodactylum is the better one, but I know that there are many groups in everywhere that are settle other organisms like Ketoceros that maybe in the short term they will become model organisms, let's hope, and that could be amazing too. Yes, but for now I plan to do it very simple. Yeah.
And in plants also, so you also mentioned plants?
Yes, for plants, I have some idea, but in my experience, for example, tobacco is a good system, easy to transform, big biomass, and also a crop, not a very, well, yes, it's a crop after all, where Arabidopsis, I see it more like an ecological, I know that it's not like this, but it's a protein from nature, so it can be nice as an ecological system, but I think it can be much more simple to solanaceous like tobacco, for example, or other one, yeah.
Okay.
But I am, let's say, biased by my experience, but I think it can be adapted to, yeah, whatever.
Thank you.
Other questions, please? So I was very interested by your finding of clustering of aggregates of cells of Synechococcus that was generally considered a free phytoplanktonic organism. So does this have implications for other organisms? Are planktonic cells now so free living in general or more living in aggregates? What do you think about that?
Well, according to what I have seen in the microscopy data from Tara Oceans, I didn't find something as in the cases of these Picocyanobacteria. So maybe this is a particular case. Yeah. The important thing about this is that in general we tend to see, for example, the biological carbon pump is we tend to consider very big organisms like large diatoms. But in recent years, people doing sampling in the deep ocean or a very deep waters, they found by by molecular methods signal from Synechococcus. And at that moment, it was not very clear how they end up there, and I think this is a good mechanism to explain that. Yeah.
Very nice. Okay, if there are no more questions, thank you again, Juan, for your wonderful talk.
Thank you.
And welcome here.
Cristina. Sí, sí.
¿Te puedo preguntar una cosa?
Sí, sí, por supuesto.